Good evening, everyone. In fact, on beautiful evening of these festives, we all are here to hear from a prestigious speaker. And as always, we have program director and moderator, Professor Dr. M.B. Agrawal, sir. He doesn't require actually any introduction, but for the beginners as well as my fellow colleagues of first year residents, whether it is DM or DNB, just a small introduction of sir. Professor M.B. Agrawal, sir, is a professor and head of Department of Hematology, Bombay Hospital, Institute of Medical Science, Mumbai. Sir is a consultant hematologist associated with Lilavati Hospital, Bridge Candy Hospital, Mumbai. Sir is the president of Mumbai Hematology Group, past president of Indian Society of ISHBT, as well as sir is institution in himself. Sir is inspiration for me as well as for many and teachers of the teachers. Sir, India is fortunate to have you here and who has enriched us a lot, sir. And collaborating with many stalwarts in hematology across the globe and bringing them on virtual platform like today. Right, sir has been awarded as the inspiring hematologist of India by prestigious media like Economic Times. Sir has been awarded Guru Dronacharya by Indian Academy, Leukemia Academy. So, sir, over to you with this small introduction of yours.
Thank you, Hitesh, for those kind words. And good evening to one and all. This evening, we have Dr. Amrit Kaur Kaler from Mumbai as our guest speaker. She's going to speak to us on the very basics of the molecular hematology for the beginners like us who want to understand everything about what molecular hematology means. This webinar is brought to you by Mumbai Hematology Group. It is supported by INTAS and it is managed by Perfect Square. I'm grateful to Dr. Manoj Kumar, Hitesh and the team from INTAS, Yash Kalpesh and team from Perfect Square, Executive Committee of Mumbai Hematology Group, our guest speaker, Dr. Amrit Kaur Kaler today, and the discussants who are themselves eminent hematologists and medical oncologists. And I'm sure all of you will benefit in learning what we have planned today. And all you participants from various parts of country as well as the globe for sparing your Saturday evening, afternoon or morning to understand these subjects' basics. Tomorrow morning at 11:30 a.m. Indian Standard Time, we have Dr. Delphine Rie from Paris. She'll be speaking to us about a very important subject, and that is asciminib-based treatment of CML. As you know, asciminib has been just launched in India just a couple of weeks ago, and therefore we need to know everything about this molecule in the treatment of this commonest blood cancer, chronic myeloid leukemia. Discussants, of course, are very important personalities from India and around. We have Dr. Akanksha Garg from Ahmedabad, Dr. Ankit Batra from Dehradun, Dr. Ashraful Haq Chaudhary from Dhaka, Dr. Debadrita Ray from Bangalore, Dr. Fahima Hassan from Prayagraj, Dr. Nin Mia Thanda from Myanmar, Dr. Subrapanyam Kanan from Pune, Dr. Madhura Dinekar Deshpande from Pune, Dr. Manu from Sri Lanka, at present in UK, Dr. Mohammad Mir Hussain from Srinagar, Dr. Maushmi Suryavanshi from Faridabad, Dr. Nikhil Kumar from AIMS, Dr. Pritish Chandra Patra from Bhubaneswar, Dr. Priyanka Chauhan from Lucknow, Dr. Rahul Arora from Nagpur, Dr. Rizwan Akhtar from Lucknow, Dr. Ruchi Gupta from Lucknow, Dr. Yatendra Parashar from Lucknow, Dr. Yi Moon Thant from Myanmar. And now it's time to introduce our guest speaker to you all. Dr. Amrit Kaur Kaler. She's consultant molecular pathology and genomics in the Department of Lab Medicine at the prestigious Kokilaben Dhirubhai Ambani Hospital, Andheri, Mumbai. She's trained in clinical genomics testing of hematological malignancies as well as solid tumors. One of her current interest areas is connecting morphogenomics to precision oncology. Her top research area is immunotherapy along with new targeted therapy, which is changing the course of cancer. She's skilled in handling lab operations across department of histopathology, hematology, and clinical pathology. She has over 50 papers in national and international journals. She's co-author of Postgraduate Pathology book. She's recipient of multiple awards in the area of pathology and leadership. She's also a regular speaker in numerous national and international conferences and seminars. She's very passionate about women empowerment and helping women in rural India. I just said she'll be lecturing on ABC, that is Basics of Molecular Hematology for the beginners like us in clinical hematology. Well, thank you so much. And now I hand over to her for her lecture, followed by discussion.
Thank you so much for that introduction, sir. In fact, we are very much privileged that we have you as our teacher and a guide to show us the way forward and let us know the applications of the molecular pathology in the field of hematology. In fact, whenever I come to the biannual conferences that you held, it is like amazing that how the precision oncology is changing even in the field of hematology, and we have so many targets which are coming up, and it is showing us how the cancer maybe in the next decade or the next 20-30 years we should be able to have treatments in which we could change the course of any disease that is has happened. So it's rightly said, when there's a disease, there's always a solution. We just need to look into it and find it and give it to the world. So I think this is what motivates us, and you have provided us a wonderful platform to explore that. And even when we listen to all the molecules, we are also amazed every day. Whenever we report, we see that this is a new molecule, and wow, okay, this is going to give a different prognosis and a different prediction in this patient based on the other parameters in hematology. So thank you so much for giving me this opportunity. And I think when I say ABC of the molecular hematology, I will be talking about the basic techniques that are being used in this field, and how to utilize them in the best of the clinical practice. This is my main agenda of the talk. And what are the basics of genomics which we need to understand, because when we are interpreting the report or when we are reading the molecular report, sometimes we are confused that how to take this forward. And this is something which I need to talk. And in the restrained countries like India, in which the patient is not able to afford so much of resources, in that particular situation, how to choose which particular test, this is important to understand. And what are the sample requirements, and how to take this specialty forward for the use of both the molecular pathologist and the molecular hematologist like you, sir. Thank you so much. So I will just start my lecture. I will just explain what is a central dogma. So central dogma is nothing that I think we have learned all in our 12 standards, that DNA is converted into RNA, and this process is known as a transcription, and then the RNA is translated into proteins. And now the term metabolomics has come based on the breakdown of the proteins, whatever is formed, like sugars and nucleotides. Now, the conversion of RNA to DNA is also known as a reverse transcription. So this is the basic central dogma of any genomics, whether it is a solid tumors or whether it is a hematological malignancies. So when we go to the next, since karyotyping and the translocations and even the chromosomal instability, genomic instability is very common in hematological malignancies, and it is still just picking up in the solid tumors. So we need to understand what is a balanced rearrangement, what is a fusion, what is the imbalances, what is a cryptic fusions. So these are the certain terminologies which we need to know as we go forward. So when there is an exchange of a product from here to the other chromosome and it is a balanced rearrangement, then it is known as a chimeric gene. And chimeric gene is sometimes also known as when there's a one exon, is it goes and binds to the other exon. Exon 2 binds to the exon 16. But if sometimes exon goes and binds to the intronic variant, so it leads to the unstable fusion protein. So when this is an unstable fusion protein, it is usually not easy to catch up on the smaller techniques like karyotyping, and sometimes we are able to pick up on fish, and sometimes on NGS we are able to do that. But if there's an imbalance in, a loss from one goes and gains in the other one, and there's a loss here, we call it as a chromosomal imbalance. So we call it as trisomies and monosomies. And this is how I will take you forward towards a triplet code. So when I say a triplet code, DNA is basically 4 bases, AGCT, which is converted into a triple code, and this is converted into a protein. Now, the types of the mutations is important to understand because we deal with the substitution, we deal with the insertion, and we deal with the deletions. Substitution, we know that it is A is converted into T, and we say insertion, there's an extra nucleotide which goes into that. And when we say deletion, one nucleotide is always deleted from there. So just 1 second. Yeah. So when we say targets for molecular diagnostics, we need to know what are we dealing with. Are we dealing with a fusion? Because if it is CML, we have BCR-ABL. So we need to choose the fusion protein. Or we are dealing with a single nucleotide mutation like MYD88, or we are looking into a 5q deletion, which we see in MDS. So we need to understand what is the type of disease we are looking, what is the type of mutation or a chromosomal translocation or a deletion we are looking. So based on that, we will choose the technology what we require. So what is the starting material we need? Do we need to run DNA or RNA? Or do we need to run both? Or what is the source of the tissue that we need? Is it a fresh tissue, or it is a FFP block if it is a lymph node biopsy? What is the type of abnormality we are looking? Because if you're looking at CLL, or if you're looking at MDS, we are actually looking at numerical abnormalities, monosomies, trisomies, and polyploidies. Polyploidies are usually HER2 mutations. So when we are looking at all these mutations, we need to decide what we need to focus on, what type of technology we need, and how do we need to proceed. So we need to differentiate. If it is a DNA based, we will be looking into single nucleotide variations, small insertions and deletions, or even duplications. Duplications means one chromosome is getting into 2 more copies. So now when we talk about gene fusions, we are seeing here the 2 is combining with 16. With the result, we are getting a fusion protein. So this is a heterogenomic alterations which we see here. Now in JAK2, we are looking at a single gene mutation. In Intels, we are looking at FLT3, ITD, NPM1, or even CALR. In CNVs, we are looking at 5q deletions. And if it is structural variations like PML RARA, so these are the different types of abnormalities that we see. This is just what I want to explain. So what are the methods? If we are looking at a single gene mutation, RT-PCR is the best way to deal with it, and even Sanger. And Indels is something picked up by RT-PCR. RT-PCR can also pick up fusions and even the clonality. But when we look at FISH is able to pick up only fusions and CNVs. So this is important to know. When we look at arrays, we are actually looking at copy number variations. And if it is NGS, it is looking into all the different types of abnormalities, except the copy number variations, we might have to go to MLPS. So let's have a look at the different advantages and disadvantages. I think this I will discuss as it goes along. I think this I have already introduced. So what are the clinical applications? The applications are, how do we diagnose it? How we classify a hematological malignancy, and how do we need to prognosticate the disease? Yes. Are we looking at monitoring? Yes. Or are we looking at a targeted or a precision oncology in hematological malignancies? So in that case, I will just start with the DNA extraction. And I think I don't need to get in deeply into the extraction, but I'll start with the cytogenetics. So cytogenetics means karyotyping. It is a widely used for the dividing cells, which we capture after culturing them. And we look at the amplifications and the structural variations like the translocations, deletions, and inversions. So in this, we collect a heparin-based sample. We, put a colchicine on it. We culture the cell, and then we stop the division, and we harvest the cell, and then we isolate and we observe under microscopy. So in this karyotyping, the turnaround time is 1 to 3 days. But the limitations of this technology is it is only for the live cells. There's a low resolution. 5 to 10 MB is a low resolution, and there's a low sensitivity because the number of cells that you capture are very limited. So that is why the sensitivity is 5 to 10%, but it is widely used in all the hematological malignancies. Next is the fish principle. In the fish, again, we prepare the sample. You definitely don't need a dividing cell, but you can use both cultured cells also. But then you use a fish protocol in which you denature, you hybridize it, then you put a DAPI on it as a counter stain, and then you see it under fluorescence microscope. So the type of probes is very, very important. What are you dealing with? So this is a break apart probe. You see the cells which are fused are now broken apart. It means that the name is derived from the vision, the picture, the type of picture that you see on the slide, on the microscope. So if it is a dual fusion in which the cells are separate on the chromosome, the centromeres are separate, but if there's an abnormality, there will be a fusion. So this is how you see in BCR-ABL and PML-RARA. And enumeration is usually seen in HER2 amplifications. So you can see this is a P53 probe. There is an abnormality in which one pink is missing. It means P53 is deleted. So it is 2 green and 1 red. It is a confirmatory diagnosis that this is a deleted P53 that we see here. Then in the PML-RARA, you can see the fusions here. 1 green, 1 red, and then you have a mixed color. There are 2 fusions and 1 green and 1 blue. So it means as a PML-RARA is positive. Then you have to talk about the number of cells in which they are positive, and it can detect the cryptic abnormalities. And yes, you can identify the specific targeted regions. Coming to the DNA microarrays, they are used for the small unbalanced abnormalities, but do not do the balanced translocations, which is very nicely seen in karyotyping. So every technology will give you a tubic view of the abnormality sometimes. So that's why you will have to mix and match to understand sometimes how to go about. Now, MPN first used to be treated like you do a fish for BCR-ABL. If it comes out to be negative, then you do a reflex MPN. This is how sometimes in many of the places it is being used. But now the panels have become like MPN panel. At one go, you go and see if there's a major, there's a minor, CALR is present or not, and MPL gene is present or absent. So like this, you can, instead of doing a reflex panel, you can screen the patient for any other abnormalities and, start treating the patient. So the different panels, and again, the approach is based on the different oncologist, based on the clinical setting. So all these are important to choose how to go about. So then when you talk about DNA microarrays, at a time, you can go into different wells, you can process different samples at a time. You can put the probes and see it under microscope. And then you can see 20 to 30% of the cells are important, and the amount of the base pairs which is seen is very, very important. It can detect up to 20 to 100 kbs of the base pairs, which is important. Now, when we talk about MLPA has been used in germline cancers in which BRCA1 and BRCA2, you see a bigger deletions and duplications. So I thought I should mention this technique because it should not create a confusion. It is not used for mutations. It is only used for deletions and duplications. And in this also, the product is formed, and then this product is amplified, and then different hotspots on the gene can be amplified and seen and measured based on that. So these are the different steps. Then coming to the PCR amplifications. When we say PCR, then PCR is very commonly said, you're making a nice bath in which khichdi, you can say, DNA template, nucleotides, stack polymerases, PCR buffers are used. And with this, you are actually dividing the DNA. You are actually opening up the two double strands, and then you're putting the primers, which are starting the initiation of the, individual single strands are even moved forward, and then there is an extension of those primers. With the result, the two strands will become 4. So both of them can be sequenced. And this happens with the help of a thermal cycler, in which, these 3 different steps are performed at 3 different temperatures. Denaturation usually happens at 90 degree, and the primers usually happen at 40 to 50 degree, and the extension happens around 60 to 70 degrees. So there's a fixed temperature. There is a fixed time, which is used, 30 minutes of PCR or 20 minutes of PCR, based on the different reactions. So we also have like 2.5 hours of PCRs also based on the different tests that we need. So basically, they are amplified to such an extent, and then you can measure the weight or the base pairs that have been formed. So you can see here, this is 400 base pairs, this is 100 base pairs, and there's a ladder which is being used. So NTC will not give you any results. So this is the, weight of the product, if it is like how much it is. So if it is 400 base pairs, and this is the 100 base pairs, so it will all depends upon the migration on the this one electrophoresis. Then this is a tape station in which also we can measure the product. Now this PCR is used in all the different techniques: Sanger, electrophoresis, capillary electrophoresis, which is a fragment analysis, MLPA, next generation sequencing, and even transcriptomics, and all different types. Now let's talk about different types of PCRs. There are many of them, but out of these, only very few are used in the diagnostic scenario, because we don't need to learn all this. We just need to know these allele specific, real-time, reverse transcriptase, and multiplex and nested, because these are the 5 PCRs, and including the digital PCR, which is used very routinely in the diagnostic setup. Now, whenever you're deciding a PCR reaction, you should think about the 2-3 things. Are you looking for a single target? Are you looking for a more number of targets? Are you just wanting to look for a qualitative, whether it is present or not present, or whether you're looking at a quantitative? How many copies are there? Like in the BCR-ABL, you want to know how many copies, 10,000 copies or 500 copies. And, when you sequentially monitoring the MRD in CML cases. So that is a quantitative PCR. That is known as real-time PCR. Then whether you're looking at a single mutation, or you want to do a sequence variant detection. So it means you are looking for one or multiple. So how do you want to get it detected, and what is a threshold? So most of the PCRs will have around 19 to 27 CT value, in which you can say it is positive and negative. And sometimes when you are looking at a borderline cases, suppose in JAK2, it is a borderline. It is 27 CT value is the cutoff, and you're getting 28. So you might want to repeat it, or you might want to think this is a somatic mutation instead of a germline mutation. So you would want to monitor the patient or follow up with the patient. So these are the different types of the things you want to see in the lab, that how you could help the clinicians, the oncologists, in deciding what they are looking at. How do you want to validate the assay to understand how do they want to approach a single case? So this is how you can suggest the clinician that how to proceed in this particular case. This is very helpful sometimes. So when you're looking at allele specific means you're looking at one single gene, example V67F, which is exon 14. Sometimes the request comes, it is only exon 12 that we are looking at. So exon 12 is most commonly done on a Sanger sequencing, while V617 is easily applicable on the real-time PCR. And sometimes you're looking at a BRAF for hairy cell leukemias, and sometimes HPP. So or you're looking at a bigger panel. But if you're looking at a product, want to measure the product, if it is like 400 base pairs or 200 base pairs, PCR is not the technique. you are looking at a single allele which is abnormal, and you want to know it is present or absent, allele-specific PCR, also known as ARMS PCR, if there are multiple points, multiple targets are there in the panel, that is known as ARMS PCR. If it is like single gene, then we also call it as an allele-specific PCR. So I'll just explain. This is a sequence which we see in JAK2. This is the wild type in which you see a G here. But if it is mutated, you will see a G is replaced by T. So you have a JAK2. When GTC is there, you have a valine. That is a protein which is coded. So V, and at the codon 617, now this V is replaced by T, and it is replaced by phenylalanine. So that is why it is known as F. So this is how is the annotation of that particular mutation is done. So this PCR is allele specific. It is qualitative. It will only tell you an information if it is positive or absent. So you can see here, this is a G and this is a T. So this is a wild type. This is a mutant type. So in an assay, you will run a normal primer with a wild type allele, and then you will have a mutant allele. So the PCR will show you a reaction of both the alleles if the mutant is present. Amplification of both the alleles. But if it is only a wild type allele, only the wild type PCR will, the curve will come. But if it is mutant also, both the curves will come. So then you will calculate the CT value based on minusing with the standard PC, and then you will give the positive results. So in this, you can see G, this is the common allele, this is a T, and this is the G. So this is how you report it. When you call it as a real time means you want to measure the number of copies. This is particularly used in MRD monitoring. And CML, I think it is very, very commonly used. PML RARA is also something. RUNX 1 is also something in which real time is being used. So it is a very, very, very good technology. So you put a forward primer, you add a probe to it, and you put a fluorescence here, and you add a quencher here. So if there is a mutation, the probe will attach. If there is no mutation, probe will not attach. Now, if there is a mutation here, probe will attach, the sequencing will start, and when the sequencing will start, quencher will release the fluorescence, and that fluorescence will be captured by the laser, and that will give you the peak over there. So the more the number of peaks, the fluorescence getting emitted, the more the number of peaks getting formed, and then you can pick up how many number of copies are there. So this is how a real-time PCR works. Now this is again the exponential increase you can see here. This is 8 copies, and by 15 to 20 copies, you are able to see your curve here. So PCR in fact is a very, very easy technique. You can easily monitor the assay in a day's time. Probably the DNA is extracted a day before. Within half a day, or you can actually release the report. You can tell the clinician is positive and negative based on that. Now, what is reverse transcriptase? Whenever you see a RT-PCR, don't say this is a real time. This is a reverse transcriptase. This is something mistake which I think many of us, even molecular pathologists, we have a doubt in the beginning. But then this is why this was discovered, because when we say mRNA transcripts, we want to convert them into cDNA. Okay, so when we are reversing that, we want to, reverse, instead of DNA to RNA, we want to convert from RNA to cDNA. So we need to use a different enzyme, and that enzyme is reverse transcriptase, so that we are able to sequence that chimeric fusion protein. So now when we do that, then we put them into real-time PCR, and then we take it further for measuring the assay. So it is basically reverse transcriptase real-time PCR that we use for MRD assessment. Now, these are the applications we all know. AML1 ETO is something some clinicians wanted. PML RARA and BCR-ABL is continuously being used. And these assays are very, very nicely validated in the department. We run 6 monthly calibrators to understand, to validate the IS scale, so that we are able to give the correct results. So there were so many problems which were discussed and which we see on the daily basis. I can take those questions later, because the ABL1 which is being used is a housekeeping gene. That is an indicator which will tell you how healthy is your assay that you are using, how healthy is the patient sample, because transportation usually kills the mRNA, which has to be converted to cDNA. So the quality of the sample is measured by ABL1 copies, which I think I will show you in my next 1 or 2 slides. So this MRD monitoring was again very commonly used because of this great history which was created a long time back. So now this is the report which we release here. You can see the BCR ABL copy numbers are 15. Now the ABL copies are around, you can measure 3,07,809. So when I look at that, we have another reports which says, okay, we have, 5 copies, and ABL copies is just around 10,000 or 20,000 or 30,000. So that is the time I know that the sample is degraded. So whenever you have 2 reports in which says the ABL copy numbers is 3 lakhs, the other copy says 30,000, and that is the time we should think that the one which have 3 lakhs has the sample which was retained properly and it has been processed properly. So this is how you check the authenticity of the lab and running the assay, because as this value decreases, your MR will decrease. Your IS scale will get disturbed. So suddenly you will see a higher IS scale. So this is how we check the quality of the reporting in any laboratory. So now we have a lot of assays which have come up. In a single acute leukemia, I say there are copy numbers which are available. One panel will have 8 to 10 different fusions which we can detect in one go. So you have multiple targets. You have 8 different fusions in one PCR. So we call it as a multiplex PCR. So either you run it on a fragment-based electrophoresis or you do it on by real time. So the next slide. Okay, so this is again, same indication. Now this also in PCR APL, we have E14A2, E13A2, E19A2, different types of major, minor, and micro, which we measure depending upon the assays. So those will be qualitative assays. If we are measuring just one allele, then we call it as a quantitative, just for the MRD measurement. And now these are different applications. The nested PCR is also used for IRMA. So what is a nested PCR? In the nested PCR, we have 2 sets of primers. We first sequence one set of gene, and then when we have sequenced it, we go to a smaller targeted sequence which we need, and then we add a second set of primers to amplify that part, and then we are able to get the results. This really increases the specificity and increases the accuracy of the results. So this was this technique is being used for IRMA reporting. Then coming to the digital PCR, it has a good applicability, because again, a digital droplet means you are actually having the bubbles, you are creating a reaction which is uncontrollably high, and it is the sensitivity is 10⁻⁵ in this particular case, and it is being used in solid tumors also, and hematological malignancies and all the fusions and mutations. So I think NPM1 is something very commonly used. It has a highly reproducible results, high precision, and multiplexing is also possible for, depending upon the type of ddPCR that we used, it can be 4 or 16, and the results can be measured at the same time. It's quite easy, only the separating the ctDNA is something which needs to be be very careful, because we need to have those Streck tubes which are very important to store the ctDNA. So this was all about the PCR. Then we talk about the fragment analysis. So until now, we were just looking at point mutations. We were looking at SNV, single nucleotide variations. Now we are looking at the product, because there are some mutations like FLT3, like NPM1, like CALR, in which the product is formed, the big product is formed, the big deletions will happen. So that product we need to measure and understand how big is the deletion. So now this in the FLT3, I think the ratio of the mutant to wild is important, or was important, because now I think we have changed our practice to NGS now for that. But before that, fragment analysis was really, really helpful in understanding that part. So again, the technique is very simple. We have 3500 DX capillary electrophoresis provided by Thermo Fisher, which is being used, I think, in all the laboratories. Again, the DNA is extracted, probes are attached, amplified, and then the product is measured based on the migration, based on the electrophoresis. Similar technique that we use for hemoglobin electrophoresis. The faster will migrate quickly. If it is 100 base pairs, it will migrate quickly. If it is 250 base pairs, it will be slow. So based on that, we can measure the product. The bigger product will be behind. So and the smaller product will be in the front. So we can understand. We know that FLT3 has, suppose let's say 350 base pairs is a normal wild type of product. But if it is mutated, or we can say, insertion is there. So it will be from 250, it will become 290. When it is 290, it will migrate slowly. So it will be left behind. So this is how we measure the size of the product. And the technique is the same in fragment analysis as the hemoglobin electrophoresis, and the name is different and the machine is different. That's all. So this is the 3500 DX. Again, they use the cathode and anode type of a thing. The shorter fragments move fast, the longer ones slowly. And this is the, product which we see in the gel electrophoresis. But when we look at the final report, we get a peak like this. So this is a wild type in which I see 167 base pairs, and this is 171 base pairs and MPNG mutation. And so I see a 4 base pair, insertion in this particular case. So this is how we diagnose it. If we look at FLT3, this is a wild type 328, and this is 358. So I can see a 30 base pair insertion in a FLT3 gene. And then based on that, I'll calculate the ratio. It is 0.8. And if it is more than 0.5, it is definitely carrying a bad prognosis. Now, this is the CalR, in which I had a 6 base pair insertion, which is nicely seen. So another application of fragment analysis or a capillary electrophoresis is the chimerism, which is very commonly used in stem cell transplantations. So it is based on STR fragments. So the STR fragments are nothing but 1 to 6 base pair fingerprinting genes, base pairs, which is specific for every patient, and they are present in the non-coding regions. So we take the sample from the donor, and then we also take the sample from the recipient. We run it on that. We do the STR markers which are being used. Every laboratory has a different markers. Our laboratory has a 9 markers, STR markers that we use. And at least 3 of them should match between the donor and the recipient. And after they are matched, we tell them the name of the markers which are matching. So those 3 or 5, usually 9 out of 9 happens, but 7 to 8 is something which usually matches in the same relatives, but at least 3 should be matching. And then after that, we give them the report. And after the transplantation, we do look if it is like, initially, if it is like base pairs of the recipients are there, and then the donors are there. And gradually, as the transplantation is happening, the donor STR fragments will be replacing the recipient STR fragments. So with time, you will be able to see that donor has completely replaced the recipient. So we keep on monitoring it, immediately after the transplantation, then 1 week, 10 days, and then based on if there is no rejection, I think then it becomes 1 month and everything. So this is how we monitor the stem cell transplantation in hematological malignancies. So this is one report in which we found that 4 informative markers were there, and the recipient marrow is replaced by 98% of the donor status cells. So it is very, very clearly it is written, it happened 100%, 99 to 98%. So like this, if it is stabilized, then I think the frequency between the different testing becomes slightly lesser. So I think it has a good reports. So I think I thought I should give you a break. I think DNA has lot of secrets in the life, and I think we need to get more deeper into it to understand and take this to the next level. So the next part of the talk will be on Sanger sequencing and next generation sequencing, because they both are related, and how the Sanger, came into picture, and how many years it took to sequence a human genome. And now it is like so easy. The minimum time taken for NGS is like 3 days. You can finish the wet work, and the analysis can happen in the next 3 days. So it is 6 days. So it took years for them to understand the human genome by Sanger sequencing. But it's a beautiful technique. I promise you, it's so good. Out of all these techniques, Sanger is my favorite technique, in which the chances of error are high. But when you look into the codon, the nucleotide change, when you look at all the sequences, it's really, really interesting. So basically, we are looking into ATGC. These are the 4 nucleotides. And if they get replaced by any other nucleotide, we need to understand, is this pathogenic or it is not pathogenic? Is it just a polymorphism, or is it giving any significance to that particular patient, or does this mutation been reported in the databases, or is this carrying any significance or not? Because if it is a known mutation, you will be able to report it right away. But sometimes you do get novel mutations on Sanger sequencing. So you have to be very, very careful on what you're reporting, what does that mutation is meaning to it. The difference between Sanger and the NGS is Sanger will give you information on a small part of the exon. It cannot sequence a whole gene, because a single exon might be 200, maybe 100 to 250 to 400 base pairs. So you cannot have such a long sequence, or you cannot sequence a single gene. There were times in which BRCA1, BRCA2 were done by Sanger sequencing. It used to take days, and interpretation used to be very difficult. But it is for smaller regions or hotspots, like we have a PIK3, or in the hematological, we have MPL gene, which is sequenced because we know there's a typical mutation and we want to see whether it is there or not. So it is, the application is totally different. So sometimes if you have a novel mutation on NGS, you want to confirm whether this is actually there or not, and it is pathogenic on NGS, you confirm it by Sanger sequencing. So this is how Sanger sequencing is being used. So it is very simple. You are putting nucleotides, but you're also using ddNTPs. And the usage of ddNTPs is it stop the sequence. So if dNTPs attach, the sequence will grow forward. And if ddNTPs attach, the sequence will stop, and the fluorescence will, it will fluorescence, and this will be picked up by the by the sensor, and then when it is picked up by a sensor, it will give you a sequence of GACT, GACT. So in the capillary, you will get a band or a curve. In this, you will get sequence like this. So, and this is measured in a chromatogram, and then we, compare it with the normal reference gene, and then we see whether there is a mutation or not. So this is, I think, the same thing which I was trying to tell. ddNTPs are attached along with that, and this is how the product is formed. Wherever it stops, this fluorescence, this is detected by a detector, and the data analysis is done here. So this is this picture I really liked. This is wild type, and in this you see that a C is the normal sequence, and different nucleotides are given different colors. So the C is a blue color here. So C, when the C is present here at 677C, it means it is a wild type. Wild type means normal. Now, if it is replaced by a red, which is T, so there are 2 peaks. So, every gene has 2, one from the father, one from the mother. So he has a normal allele, wild type allele. He also has a mutant allele. So it means he is heterozygous. There are 2 peaks which are seen here. But if I look, there is no blue, there is no C, and there is only red, which is a huge peak. It means that the patient is a homozygous for T. So 667 homozygous is pathogenic, and it is associated with thrombosis and other conditions related to that. Now, since it takes a lot of time, Sanger sequencing, when we do, even though it's so interesting, it takes 3 days of long time of cleaning and amplification and sequencing and the tray. Then it depends upon your having a 8 sample tray or 24 sample tray. So based on that, array. We just manage it in the laboratory. But but with the if we talk about the accuracy is definitely 99%. But as we are moving forward in this world, we want accuracy, which is 99.999. So we want to sequence it further to increase our accuracy and, shorten our time. And we want to have a high throughput. We don't want to sequence a single gene. We want multiple genes. We need multiple hotspots. And, with the requirement comes the new technologies, and we are able to, sequence, and we're able to help the patient by reducing the turnaround time and reducing the cost. Yes, there are multiple factors which come into picture. One thing I always say, that why you want to do just a single gene to save the cost of the patient? Why not do multiple genes, at a time, so that you have a clear picture of the biology of the disease and what you're dealing with and how to deal it forward. So for people like me, I think NGS is the way forward, but definitely Sanger has its own advantages. It is a very, very also accurate, but it definitely, if you're dealing with a somatic mutation. So it's very important to understand what is a somatic and what is a germline mutation. Somatic is acquired. Germline is when it is present inherited, when you got it from your parents. So it means 50% of your body has the mutant gene. If you got it either from the father or from the mother, if you got from the father or the mother, 50% of your cells will have that mutation. So even if you treat, it will not disappear because the cells are inherently producing that type of mutation. So that is why the BRCA1, olaparib, and everything came forward to, antagonize that. So if you're dealing with the germline, you need to have sensitivity of the Sanger is just more than 20%. So if you have a dealing with the somatic mutation, you might miss it on the Sanger sequencing, and which we are able to pick up on NGS. So this is what I'm trying to say, that Sanger also has its own limitations. So when we talk about NGS, we have different types of sequencing. We have a targeted whole exome and whole genome sequencing. And in the targeted, we have, we can define the number of genes, whether we want 80 genes or 50 genes or 200 or 500 or 1000. So we have panel now 2000 genes also, which is hybrid capture and which is hotspot mutations. Then we have a whole exome and we have a whole genome. Then we have one in between, clinical exome. So clinical exome is around 8,000 genes, and this is whole exome is around 20,000 genes. And then we have a whole genome sequencing, in which you are even sequencing the introns and other things which are not the coding part of the DNA, but sometimes people want it. And there are some conditions in which you do have some intronic variants, splicing variants, which are changing the whole course of the disease. And I think now the lot of interest is also based on that. So that is a time in people go for whole genome sequencing, but it is not a part of the routine diagnostic test, which is required for treatment and for managing the patients. So this is, again, as I mentioned before I go forward, hybrid capture and amplicon-based method. When I say amplicon-based method, the whole of the gene, it gets amplified. So the amount of data which gets sequenced. So the amount of data which is generated is quite huge. So that is one thing, but the whole gene gets sequenced, and you are actually sometimes you get a false amplifications, but the chances of missing a gene is lesser. So when you talk about the hybrid capture, so on a gene, you have multiple spots, you have primers designed to detect only those hot spots. And then you do the PCR amplification, and then you make a library, and then you sequence it. So there are specific hotspots within the different genes that is known as a hybrid capture methodology. So initially, Amplicon was generating a lot of data, and bioinformatic analysis is something which is, carries a huge amount. So so that was not available. So that I think hybrid capture is more useful in the clinical practice, because when you know this is the mutation and this is what you want to know, and then you want to treat it further. So I think in clinical practice, hybrid captured ones are better useful. But it also has its own limitations that we can talk about. So what are the basic steps of NGS? I just don't want to get into the detail. We do the DNA extraction. That is a normal thing. We check the quality either by tape station or by, electrophoresis. After that, we do the library preparation. We add the adapters and we do add the indexes. When we do that, we are actually, putting identification mark on the samples, because in one run there will be multiple samples. And then you will put the primers on the particular hotspots that you want. And then you will do for hybridization by overnight incubation or by 4-hour incubation or 10 hours based on the different types of panels that we use. And then after the next morning, you clean the product, you measure it, you denature it, and then you, put it on a sequencer, whatever sequencer you're using, Illumina or the other Thermo Fisher you're using. So you put it there. And I think sequencing goes on for, based on the different type of sequencing. We have Illumina, so it goes on for 24 to 36 hours. And then the data is generated, and that data is being transferred to a bioinformatics pipeline, which is being used for demultiplexing and taking it forward. So this is one picture in which I wanted to say this is how the sequencing happens. This is one getting and forming an umbrella shaped here. So, and then this is the basis. This is the basis for the next sequence. And after this gets sequence, and this is the basis for the next sequencing. So then there were different questions. The basic sequence is lost on the way. So the chances of errors are high. So to reduce that, then they had a Fred score known as Q30, which should be more than 90%, 99.9%. Or, Q40 also now has come in Illumina just to reduce the number of errors. So this is some technology which we need to know. And then, of course, this is the bar coding, which I think I've already explained, but the product has to be 350 base pairs. This is important. 350 base pairs should be there if you're measuring a hotspot, just to prevent the errors on the sides. So I think bridge PCR, which I've already told you, the cluster formation happens on the flow cell. And when there's the cluster formation, the fluorescence, the ATGC is releasing the color, and this is being picked up by the camera. And when this is picked up, and this is how the sequences are formed inside that NGS machine. Now, let's have the bioinformatics analysis, a little bit about it. So there are different reference genomes. I just wanted to cover this first, because when you get a bioinformatic analysis, you are doing a primary reporting, secondary reporting, and tertiary reporting. In the primary and the secondary, you are just doing the VCF file. VCF is variant call. You're doing a variant call by demultiplexing and then converting them into a single sequence. And then single sequence is, aligned with the reference sequence. And when you're doing that, when you know the reference sequence has CTGC and your one is CTAG, or the sequence is different, this is how you are the system is able to pick up. The analysis is designed in such a way that it will be able to pick up that mutation. Now, the two things are very important. One, quality parameters. Coverage, whether the hotspot of 350 base pairs is adequately covered or not. Because when you're looking at a NGS report, this is what you have to see in the report, whether they have mentioned the coverage or not. Because sometimes they say, okay, this hotspot was not covered, and you're looking only for that hotspot. I had this experience because we don't do whole genome sequencing, and we have outsourced one sample for EXT gene. And that is for, exostosis, that a patient presented with multiple exostosis, the whole family had it. And we had outsourced that sample. And then when I asked him, he said, Madam, EXT gene, the data was not good enough. So we were not able to report that. And that's how you miss the mutation. So coverage is something very important. Then depth is important. Depth, why it is important? Because if you have a depth of around 5,000, let's say, okay, and you're able to see it only in the 5, reads, the 5 out of 5,000, it will just come to 1%. So 1% is not significant. So some people reported, but we have a cutoff of a somatic mutation reporting at 5% and more. And for the germline, you don't need to go to 5,000 depth. You can reported at least 100 depth. And at 100, if you are able to see it in the 40 reads, so you're dealing with a heterozygous mutation. But if you're dealing with 80 reads out of 100, it means you're probably dealing with a homozygous mutation. So this is how we see, whenever you see an NGS report, you must see, have the report mentioned about the coverage and depth of the whole run, whole sample, or that particular hotspot or that particular gene. Because we mention, if the mutation is reported at this gene, at this hotspot, we mention it below that this particular mutation is seen at this particular depth. So that gives the authenticity to the report. Yes, we have actually seen it, and this is the VAF. And this, if it is a high VAF, you need to treat it extensively. If it is a low VAF, okay. So you need to look into other fusions, which might be significant. So the reporting of fusions is again a different ball game. So you don't look for VAF, you just look for how many reads it is there. So if it is significant, if it is PML RARA, and PML RARA usually don't go to NGS, but definitely if you see a PML RARA and if it is there at 2 to 3 different reads and the patient is a known case of AML, we report it. But usually it is seen at a very high concentration. So these are the two important things which I wanted to say. And VAF is again something we use it only for mutations. We don't use it for fusions. So these were the important points. And you can see here, again, the G is converted into T, and this is a VAF. For this, it's quite higher. So probably this is a germline mutation. This is how the bioinformatics look like. This is how we see every day. So this is the reference genome. I wanted to share this, and this is the exon 11, and this T is red. So it means it has been mutated. So V617F is positive in this case with the high VAF. So before I go there are different databases which are available, which will talk about, the pathogenicity of the genes. And you can look into ClinVar, you can look into Ensemble, how these mutations are and how many cases it has been reported and how is the presentations. And even there is one known as, if you want to look into the gene, you can just type the gene and the C and P about it and look for OMIM. OMIM classifications are there. So you will get the whole history of the whole gene, in what all malignancies it is seen, how it is, whole presentations and everything will come into picture. And sometimes clinical synopsis is also there in gene reviews. So that actually helps a lot in clinical practice when you don't have, sometimes you get a novel variant and you don't know anything about it. So that's the time these websites really help. And ClinVar is also something which is very, very useful. So this is a different, I think, company, iTorrent, and this is widely used. Everybody says it is, it misses the fusions, but I see this machine everywhere, but I think it is pretty useful. And this is based on a semiconductor technology. So coming back to the MRD, real-time PCR, ddPCR, NGS. These are the three technologies based on your availability. Sensitivity-wise, ddPCR is very good, and even NGS is very good. But real-time, it's almost equally good, but ddPCR is definitely better than that. So I just wanted to conclude here by saying the cytogenetics resolution is only 5 MB, 5,000 MB. Below that, you cannot see the resolution. So cytogenetics has its own limitations. If it is fish, 100 to 200 KB. Below this, you won't be able to pick up the fusions or even the mutations. So coming here, if you look at targeted exome, even single base pair can be picked up. So that is the reason these technologies are becoming more popular nowadays. And also it gives you the information on everything. And single nucleotide variations, indels, CNVs have this little limitations. And for that, you can confirm that by MLPA or FISH. And structural variants also you can say, but for structural variants, I will suggest you go back to the cytogenetics and FISH. And loss of heterozygosity, which I think you should go back to microarray for confirmation. So whenever I report, I see if any of this is present, confirm with cytogenetics and FISH. And invariably in hematology, these will be there. The patient will be carrying this report. So it's all in the practice. So in our hospital, if we see a case of AML, I think after the acute leukemia flow cytometry, the sample comes to us for FLT3 and NPM1, and that is reported within 5 to 7 days. And then the sample also goes for a next-gen sequencing because we want to understand the prognosis, because prediction, FLT3, NPM1, we are able to take it forward. And then we want to understand what are the biologies we are dealing with, if there is anything else we need to look into for the prognosis. So this is the practice for AML. For ALL, I think BCR-ABL is something after, say, cytogenetics is reported. For CLL, I think it's majority cytogenetics in fish. And if we look into MDS, yes, cytogenetics in fish, because then nowadays their samples are coming even for NGS for MDS, because we have SF3 1 gene, which is important to look into. Multiple myeloma, again, cytogenetics and fish, but people are gradually moving towards, bigger panels, which will move forward. But I'm not saying everything will get into NGS, because every technique will give you a tunnel vision. So you need each technology is complementary to each other, just to be sure on our side that we are dealing with the right things. But in hematology, definitely CG and fish will always be there, and targeted panels on NGS will also, come along with that. So I think I'll just end up my talk saying that we all believe in the data that comes out in papers, and but I think practically when you deal with the samples in the laboratory, you see a lot of limitations. You see, a lot of troubleshooting. I think that adds on, and I think it's best to help each other out and see what works best for the patient. With that, I think I'll end my talk, and I'm open to any questions if they're there.
Well, thank you so much, Dr. Amrit Kadir. That was wonderful. I don't think you could have made it simpler than this. People totally ignorant, which includes me, also understood most of it. And maybe we keep calling you, we will digest more and more. And it was a wonderful talk, very comprehensive, and as simplistic as could have been done. You made a great job out of it for the clinicians. So
Thank you.
now we have our colleagues here. You can use the raise hand sign. You want to ask question, you can open your videos if you wish to. And the audience, if you want, you can put question in the chat box. It's a golden opportunity for you to discuss with a molecular hematopathologist. Not every day you get someone. So Nikhil Kumar, go ahead.
Excellent talk. You covered such a complex and diverse set of topics comprehensively within a limited time.
Dr. Amrit, you can stop sharing your screen so that.
Oh, yes, sir. Yes, sir.
From a clinician's perspective, I have a few questions for you. The first is regarding the sample, because in hematology, we always feel whether we should send a bone marrow sample or whether a peripheral blood sample would suffice. So, for example, in a patient with acute myeloid leukemia with significant amount of blast in the peripheral blood, even tests like cytogenetics, can it be done from a peripheral blood sample if rapidly dividing blasts are there?
Dr. Amrit, you have to stop sharing the screen. It's still.
Okay, sir. 1 second.
Yeah, please. Yeah, done. Yeah.
Yes. So this always has been a question, every time we get a sample, that the sample has come for flow cytometry, and what sample should we send for peripheral smear and bone marrow. Usually, when we look at both the samples, the peripheral smear sample will give a 1 log reduction, but then that is again based on the total number of blasts. And if you see the number of blasts are good enough, then obviously you can go for a peripheral smear, because sometimes bone marrow is difficult. But I would suggest, if it is an initial diagnosis that we are making for the patient, then definitely go for a bone marrow. But if you're doing an MRD, for a patient, minimal residual disease, I think peripheral smear is as good enough. And then sometimes it is suggested that post-induction and post-consolidation, also the samples in the post-induction, you can go for a bone marrow, post-consolidation, you can go for a peripheral smear. But usually there is not much of a difference if you have an adequate number of blasts in the peripheral smear. But in the initial diagnosis, yes, you have to go for a bone marrow.
And my second question is regarding kinase domain mutation, KD mutation study in CML patients whom we are planning for a second line treatment. In this setting also, would a peripheral smear be, I mean, a peripheral blood sample be as good as a bone marrow sample?
No. I think you should go for a bone marrow because we have seen patients missing their mutation on a peripheral smear. This is an important decision that has to be made. I think bone marrow.
And my third question is, as NGS is becoming more and more common in acute leukemia, that we do it for all the patients, do you see going forward whether we can do NGS on a lymph node biopsy sample in DLBCLs and lymphomas?
Yes, very much.
On a lymph
On a lymphoma cases, right. Yes, I think there are a lot of panels which have come up, and there are a lot of studies which are coming up. This is definitely a way forward, and I think BCL2, BCL6 c-MYC, we have to go much beyond than that. And there are certain panels which I was exploring, which because I also wanted to start some work. I wanted to do some work on lymphomas, and there are some panels available with Thermo Fisher and even Illumina. So I think we should sequence. See, the problem happens, we don't sequence it, we don't know it. So at least
true, absolutely.
yes, if we start sequencing, we will understand that how important it is to, maybe there are some markers which are significant for the patient. Maybe we know the biology, which is better. Because if we are focusing only on the 2 or 3 markers like BCL or these ones, we are just focusing on one technology. We don't know what are the other mutations and copy number variation which might be there. So
Absolutely. Because especially in DLBCLs, we have seen patients, 60-70% of them respond beautifully to the initial therapy. But we get a small subset of 10-20% whom no matter what treatment we give, they don't respond at all.
yes,
So we very often wonder whether all DLBCLs are the same and whether we need to go beyond BCL2, BCL6 and FISH.
yes.
But is there any lab which is offering this facility, the NGS, on a lymph node biopsy on a commercial basis? Because to the best of our knowledge, we have not been able to outsource this test anywhere.
Okay, I think we should ask Dr. Nikhil Patkar if in ACTREC he has a panel on the research basis. I think that will be useful.
Can I just intervene?
Yes,
Yeah, actually for
Yes, ma'am.
DLBCL, it would be nice to make a panel. I think most of us have not, though in research, few people are already doing this kind of research. But for MCD type in ABC, actually maybe MYD88 as well as your CD79B, we are doing these two at least to because we can offer ibrutinib to these patients. But even if you do you do DNA sequencing, unfortunately, though there are hypothetically lot of drugs for PIK3CA and other types, but besides ibrutinib, I don't think there is much which you can offer. That is the main reason why these panels have not been formed. But yes, it is a good sense to do a research panel. That is one thing. And another thing I want to add to CML. So for IRMA, I think now it makes far more sense to do a NGS instead of doing a Sanger sequencing, because the limit of detection for NGS is much more. And in Sanger, if something is lesser than 20%, you will not be able to pick up. So if you are planning a study for IRMA, then I think NGS should be the technique you should be using. That's my suggestion.
Yes, ma'am. For IRMA, we have moved on to NGS. For routinely for all patients, we have moved on from Sanger sequencing to getting a kinase domain mutation by NGS. But my doubt was only concerning the sample because many times to get an additional bone marrow for sending it, especially in CML patients, sometimes it's a challenge
no,
telling the patient that we need to redo a bone marrow and send the test.
no, we
Yeah, but
had, sorry,
yeah,
no.
Please go on.
Mm.
So if you have good count, so in the peripheral blood, then it's good. It should not be a problem.
We had a case in which, ma'am, the patient was on imatinib, and the curve was like static. It was not moving. And then we thought, let's do a IRMA testing on peripheral smear, and we didn't find any mutation. And then a month later, patient came back with a log increase, and then we did a bone marrow, and we found 2 mutations, 316 and Y320. So that was quite a learning experience for us, that if you have such situation, you better do a bone marrow rather than a
But
peripheral.
the count increased. That is the catch there. So if you have a count, that is the thing.
Yeah, yes.
Thank you so much.
We'll take up one question from the audience. Dr. Amber wants to know, can we say on MLPA whether the mutation is trans or cis?
MLPA is usually, taken for deletions and duplications, and when we say deletions, in 40 base pair, deletions is the minimum. 40 base pairs and above is something which it is used for. And I think it is basically for deletions and duplications, not to quantify. I think NGS is a better technique for that.
Thank you. Another question from Dr. Hamza Dalal. In a heterogeneous disease like MDS or AML, should it be a bone marrow or peripheral blood sample for the NGS at the baseline?
Yeah, the baseline, I think I've already mentioned, we do a bone marrow. It's best to do it on a bone marrow. And then for MRD, I think we can go for peripheral smear.
Dr. Amit Khurana, your question.
Thank you, ma'am, for that wonderful talk. I have a question related to lymphoplasmacytic lymphoma. Regarding these 2 mutations, MYD88 and CXCR4, first of all, which is best, bone marrow or peripheral blood for these 2? And second, whether PCR is best or NGS is best for these 2 mutations?
I think it's the same question we are repeating. It's all dependent upon, and if the peripheral smear, it's the bone marrow which is important in this particular case. And if it is a single hotspot that you're looking at, then it is a real-time PCR. But if you want to do an extensive, analysis of other hotspots, I think NGS. Dr. Masmi, you want to add a comment on that?
So actually what we are doing is for MYD88, we have a real-time PCR. And for CD79B, we are doing Sanger sequencing because we just made it on clinician's request. And at times we also prefer fresh tissue, but we made smaller primers which can also work on FFP. So, but that saves the cost. It's very cheap. So that's the reason.
Yes.
And regarding CXCR4 mutation?
No, we are not doing that. But you can do that. It is, you can look, if you make smaller primers, you can use FFP. But if you can give fresh tissue, you can do it on Sanger, then the cost will come down very much. But if you want to spend money and you may want to be very specific, then make primers for real-time PCR. But I don't think there are kits for that available. So you will have to make your own primers.
NGS, yeah.
It doesn't make sense to do NGS for just 3 of them. It will not be cost effective. But
information will come only from the NGS panel. Unless you just want to sequence that particular gene or something. But then again, it depends upon the gene, how big it is, how many exons it is, how many introns it is, how much, it will take time for us to give us the report, because the cost versus the effort is also very important. Because nowadays, the patient, some of the clinicians, they tell me, "Madam, why don't you do this pole mutation?" So when I look at the pole mutation, the GC content is so high for me. The digestion and the primers binding is so difficult for me. GC content is high, so I would not like to see start a validation on a Sanger sequencing. I would prefer that the gene is a part of the panel and the sample is sent as a part of the panel, because the chances of missings are less, and then again, validating a single gene on a Sanger, it's a little more troublesome rather than, put everything in one panel, which I have designed even in solid tumors. For all the cancer, like lung, ovary, and everything, you put all the 80 genes in one panel and then sequence all of them and see what fits into what, and then give the report. This is how it's better in the laboratory.
Yeah, I agree about poll with Amrit. It is difficult on Sanger, but
Very
real
difficult.
time is okay. But yes, if you can do NGS, nothing like it.
And my second question is related to this NPM1 mutant AML.
Hmm.
It's always a dilemma whether to go for at the end of induction therapy
Hmm.
or bone marrow.
Hmm.
So what would be the best basically? NPM1 mutant, should we go for peripheral blood after the end of induction therapy or the bone marrow? Number one, and whether PCR would be best or NGS would be best for NPM1 mutation.
See, for NPM1, we have a PCR-based panel. So you can pick that up. Don't go for fragment analysis because that is just a diagnostic thing. But if you're doing an MRD, real-time PCR is the best. We have a true PCR kit, which is just got a good sensitivity, and go ahead with that. Post-induction, you can still go for a peripheral smear based on the number of blast cells. Okay, because you might get some of the blast cells in the circulation and you will be able to count them, but so you can based on the number of blast cells. See, we are looking at a mutant gene. If the circulation does not have blast cells, your test will become false negative. So you just want to prevent a false negative test. So better run a CBC, and if you see it's a good response, and the blast cells are very less, around 2 to 5%, and you might get a mutation because the sensitivity of the assay is 1% in the real time. Even if you have 5 ml of blood and one mutated cells is there, you are able to pick it up. But if it comes out to be negative, it's better if the patient is not responding, let's go for a bone marrow.
There is a essay available on digital PCR also. Like
Yeah.
you can, that is also good.
That is 10 minus 5 sensitivity.
Thank you.
Thank you, Amit. A question from the audience again. Dr. Kanan Ramamurthy wants to know, what is the basis of MRD by NGS? Does it differ from the normal NGS?
No, when it depends upon what you want to measure. If you just want to measure a single gene or 2 genes or 3 genes, I think single gene real time is good. If you want to, measure 2 to 3 genes, digital PCR is good. But NGS MRD is also very useful in AML cases. So I think along with the mutations, you are able to measure the fusions as well. So it depends upon the clinician, how do they want to utilize the panel. So if you're looking at digital PCR, you might measure only 1 or 2 genes, because sometimes when you have a FLT3 or NPM1, you want to measure NPM1. So that is dependent on that. So, and that's not applicable to every CLL case or CML case, because CML, it is a BCR ABL positive. So you better use a RT-PCR. So that is the reason we use NGS only for AML right now.
Yeah, and just to add, maybe if you have a new clone which is coming up, so you can come to know by NGS, and there are people who are doing chimerism by NGS. You can use that for chimerism also. So that's another advantage of using a NGS MRD.
Yes, ma'am. Just adding to that, there are so many other clones which come up. The one where precision oncology comes in, and that is where we also measure the application changes if the patient is not responding. Because when you start the treatment, your NGS gives you one result, and at the end of 6 months or 1 year, when you're not able to see the results, probably you have the mutation has evolved, and you're able to see 2 to 3 different clones, and you need to change the course, because sometimes you have FLT3, but with the course of the disease, you see IDH1 also positive. So that is a time when the patient has become resistant to one treatment. So you go for a MRD NGS. Yeah, I think that's the right thing Dr. Mosmi has referred here.
Thank you. Dr. Kannan has another question. Do you do MRD for FLT3? And if yes, then what method?
Yes, nowadays, initially, MRD, yes, we do NGS for that, and we don't do fragment for that. Yes, we do NGS. Yeah.
Dr. Mir Hussain, your question.
Yes, sir. Thank you for this wonderful talk. Definitely a very difficult subject for the clinicians. Maybe it may be easier for the hematopathologists. I just want to have a comment before going to my question. Dr. Amit had put a question to Dr. Amrit about the NPM1 mutation in AML, whether we go from peripheral blood or bone marrow. I think in that setting, we always look for the MRD status. Wherever peripheral blood will be normal, it is always advisable that we should do it from the bone marrow to look for the MRD and its NPM mutation status. Then going to my just question is, I want to know that where you put NGS in different hematological malignancies, AML, ALL, myeloma, and other like CLL, CML. So why it is utility is most beneficial, and second is, can we do away with the conventional karyotyping and then PCR and directly go to the NGS to do it? It may save some money. If we have to have an eye that we want to do NGS to look at other different abnormalities, can we directly jump to NGS instead of doing karyotyping and then the PCR?
Very excellent question, which I think this is the question I always raise in all the conferences. Why don't we do NGS at the first go? I think when we look at the clinical practice in India, we see we are limited by the cost constraint, the time constraint, which happens because NGS takes 1 month for the report. That's why the practice which is done, if it is a case of AML, go for a FLT3 by fragment in the initial phases, the report comes faster, then go for NGS. So there are multiple, even though we are trying to save money for the patient, but it does not work out. Sometimes the patient is not able to afford. So the patient is not willing. He's suggesting that maybe it's an extra cost which he doesn't want to bear. He's not able to afford the treatment which is available if the gene of anything else comes out after this. So there are multiple things which comes into clinical scenario. But definitely your question is very much relevant, and I think in the coming years, with the time period getting reduced for NGS sequencing and with the reduced cost, we should be able to implement this into our routine practice. I really thank you for asking me that question.
Yeah.
Second,
Can I just add to that? Just I have slightly different view on that. So in today's era, we cannot get away with karyotyping because your large deletions, insertions, and a lot of structural variations will not be picked up by NGS because of the targeted panel we are doing. Even with whole exome, all the areas are not covered. So in today's era, no, but there is one technology which is coming, that is optical genome mapping. And I think Amrit had that technology in her last slide, though she didn't talk about that, but that technology can go down to maybe 50 base pairs.
50 base pairs.
But still, NGS is one base pair. So maybe it can replace karyotyping and FISH, but it is maybe something like optical genome mapping and NGS right now. Or maybe long read sequencing, if it comes in few years, may replace. But today, no, it is not possible to get away with karyotyping, even if you do NGS.
we
This
can
is
say
my
it is
thought.
complementary to the
Yes.
yes.
It is
My-
not a substitute. It is, they are giving you different informations.
My second question was why we place NGS vis-a-vis AML, ALL, myeloma, CLL. So why it is utilities more in today's era?
I think Amrit, you can take it.
I think, ma'am, it's the same. AML, it has its maximum utility. And in ALL, I think the fusions are more common. So, and then it depends upon what type of fusion you're looking at. So people wants to know only a limited information. If you, there was one query, KM2A rearrangement, I want to see whether it is present or not. So they just wanted to prognosticate the case, whether it is there or not. So they just went for FISH, in the meantime, I had explained them that KM2A arrangement will give you only a single information if this translocation or rearrangement is present or not present. It will not give you an information on the partner variant. It will not tell you with what it has translocated. So that was not the query. That was not the topic which he wanted to know about it, because that is a poor prognosis whether he should go for transplantation or not. So it all depends upon how much information you want. I think a fusion panel would be a good information. If one is absent, you will know whether the other fusion is present or not. So NGS will give you a whole holistic approach to that. So I believe in ALL also it should be done. But then again, it is a belief. It is not the practice which oncologists want to follow. And also, again, it is a complementary. FISH will just tell you about the rearrangement depending upon the probe. If it is a dual fusion probe, it will give you the partner also. But then again, that specific partner, there are so many partners for KMT2A, which NGS can give you the information. But there are some partners in PML-RARA in which there are different partners, and some of the partners, they are sensitive to the treatment, some of them are not. So it is important to know which one you are dealing with. But I think that is not the first thing which people wanted. We just want to know PML-RARA, BCR1, 2, 3, which one is present and which one we need to monitor. And if there's a resistance, then NGS is being asked for. So I think specific test-based is something which we are looking at. But if you look at the role, it has a wider role in AML, in ALL. CML, not much, because the important translocation BCR-ABL is what it is defined, and majority of them are positive for BCR-ABL, and if they are negative, then you should go for NGS. I think in CLL, I think it's the translocations which are important. Karyotyping and FISH are more important, and I think that's your question. And if we talk about FISH in MDS, yes, there is a trans5q deletion, 7q deletion, trisomy 8. So these are the important things which you look at karyotyping. And then we have some mutation. The new WHO classifications also give you some mutations which have come up for the prognostication, IPSS scoring. So I think NGS has a role there as well. So this is important. And then even in multiple myelomas, I see 11-14 and 14-18, which has a treatment prediction, a prognostic prediction. But again, it's a translocation. So it can be picked up on a FISH based on your probe. But if you look at a panel, NGS panel, I think it will give you information on all the different translocations. Yes, it has a role, but then again, its applicability and the usability is based on each and every hematologist. And every technique is different. Every technique will give you a different information. So I think, yeah.
Thank you. Dr. Mir, if you want to have a clinician's reply for AML and MDS, you'd love to have the NGS panel. And for that is multiple myeloma, and CLL, you and me cannot make any use of NGS if you've got the widest panel possible available for FISH to you. So in practice, we just do FISH for CLL and and we invariably ask for NGS for AML and MDS. It's all talking from the angle of that, how you apply that knowledge to your treatment, because patient at the end of the day asks you that with this report now, what is the change you are making in my treatment?
Yes.
Okay.
Thank you, sir.
That was your question. Yeah, right. Thank you. Dr. Amrit, you want to answer question based on flow MRD?
Flow, no, I don't think so. So I'm completely a molecular.
We'll skip that. Priyanka, you go ahead with your question.
Thank you, sir. Thank you, Dr. Amrit, for that wonderful talk. So one question is related to the earlier discussion about IRMA in CML. Is there any cutoff of quantitative BCR-ABL to do IRMA and get a good result? Because sometimes we see patients are persisting on 1% or 0.9% of BCR-ABL positivity. So doing IRMA at that level of BCR-ABL, will it give a positive result?
1%, you mean the log, or you're talking about the
International ISS scale.
IS scale? 1. Okay. So you're saying if it is fixed at 1 log, whether it is 0.4 or 0.04 or 0.0, it is fixed at 1 point. Okay, so you just have to look at the graph that you see. You know, look at the graph. If it is like, going down, initially the treatment works and then it becomes static, and sometimes it starts increasing, static for more than a year or more than 6 months. So that is the time you start suspecting. But I've seen many of the patients, some of the patients, they will respond beautifully, and some of them will start showing resistance even at 6 months time, and they will start showing increase.
Sorry, I have to interrupt, but my question actually is that if suppose a patient has not achieved a major molecular response.
Okay.
Right.
In how much time?
Suppose he's a failure of therapy and he has not responded major molecular response even after 2 years, but he is maintaining a low level of transcript. He is having a BCR-ABL percentage on the international scale of 1% or 0.8%. And then if we do IRMA, if there are chances that it will come positive.
Yes.
What are the chances of it being positive? Because sometimes, many times I've seen negative reports in those patients. So is there a level at which IRMA will become positive?
No, there is no such level. But if it is a major molecular response, 1% at 2 years, there's definitely a failure of the therapy, and you should go for IRMA. You should go for IRMA. It will come out to be positive. Yes.
Okay. So predictive value is good at that level of.
Yes, 2 years of therapy. Yeah, absolutely.
Again, Priyanka, if I can add, almost 50% of the patients where your treatment has failed, there may not be any mutation.
Right, sir.
So negative result doesn't mean failure of the technique.
Right, sir. Because lately, sir, I'm seeing that I'm not getting any positive results, although the patients are not responding to therapy and they are at low level of positivity. So they are not increasing in the terms of disease status, but I'm not getting anything particular cause for that, failure. So that was my query, that whether at lower levels, will it pick up or not, the IRMA mutation?
Then again, it depends upon the cutoff of the laboratory. It will also depend upon the, what type of sample you send. It better do on bone marrow sample. And if this is the situation, and if my laboratory reports it, if my cutoff is at 5% and I am able to see at 1%, I will report it. Because it will help the patient.
Yeah, thank you so much. And my second question is that in MDS, is chromosomal microarray a better testing modality than karyotyping?
I think karyotyping is something which is gold standard here because the deletions and everything. Cryptic translocations is something which you can look into when you're talking about the microarray. Loss of heterozygosity is something which you pick up on microarray. And when we're talking about MDS, there are different things you see, loss and deletions and everything. Yes, I think you can go ahead with it. Yeah.
Because some micro deletions might be missed out on karyotyping. So
yes.
Yeah, microarray has more resolution than karyotyping.
Right, ma'am. Yeah. Okay. Thank you so much.
And I must say one thing, when Dr. Mir and Priyanka both have been asking about karyotyping. The problem of karyotyping, as Dr. Mir asked, is not the cost. It's one of the cheapest tests.
Yes, sir.
Labs, it costs almost nothing, but availability of the test. NGS, you can outsource so easily. Karyotyping, you find it great difficulty in outsourcing because you need the live cells. And most of the institutions across the country are not doing in-house karyotyping. Very major institutions are also not doing in-house karyotyping. So that is a big problem. Cost is not an issue. So in MDS, AML, you love to do it if you can have an access to send the sample across, or you're doing it in-house. Dr. Warrior has got some comments and some questions. A comment is that in pediatric oncology, we do NGS for every tumor, almost as a routine, because we learn more and understand the markers better. So that's a comment. And there is a question, is there a system where rare mutations seen in tumors are monitored for significance? We see new mutations in many cases. Any database or system used to pick them up as possible driver mutations?
Okay, in
I
pediatric,
can add my one question to that. When you said about the BCR-able mutation studies by NGS preferred over Singer, you will end up with a lot of positive information. And as a clinician, we wonder whether what is the clinical significance of that. It's very similar to Dr. Raj Varia's question.
Yes, yes, sir. So that's what pediatric oncology, again, not very much well defined right now. So I don't see many cases also in my clinical practice, and all that undifferentiated tumors. We don't, at least in our hospital, it is not being routinely used. So I don't think so. It's much in clinical practice. Yeah.
So I like to share my experience. So look, driver mutations usually have certain criteria based on which you call them driver mutation. Of course, there is a VAF, which is the variant allele frequency, and plus in which domain they are. Say, for example, it is in the tyrosine kinase domain, which is involved in the ATP exchange. So based on, there are other factors also. These kind of factors you come to know that these are the ones which are driving the tumors. But if you do sequencing, there are many things which are just passenger mutations, okay, which may not be drivers. So you need to sequentially follow the tumor up to actually know what is driver, what is passenger, and then yes, you can come to know what is the status and what the importance. But then you'll need, it is just not just one sample, maybe some sequential and other further testing is needed.
Yes, ma'am.
One more question from me about the VAF. So Mashmi or Amrit, for JAK2, are you people doing quantitative?
No.
Yes, sir, we are doing
So,
up to 1%.
Yeah, so Dr. Mashmi, if you get a JAK of say 1% VAF, what is your interpretation?
So we report it. So
So a patient
it
of
can be biallelic, it can be like in the one allelic. So those things are kept right. So if it is a known patient and you're following it up, we report it. But this is also present in the normal population. Yes, where it is dicey.
So newly diagnosed case of say hemoglobin of 19, erythrocytosis, no other features of polycythemia vera, you do JAK2 quantitative and you find a VAF of 1%. Now there is a big question mark, where are you?
That was a trick question actually.
It's a real
So
problem.
yeah, because you see that in normal population also.
There is an
So
article published just now in Blood Journal. They did the patient of arterial stroke, and they have published that paper, 11% incidence of JAK2 positivity. The VAF over there was less than 1%, 1%, 2%. 11% of stroke patients in community hospital with JAK2 positivity. It will change whole neurology.
sir, I have one thing. So this is very common, and I have some of my own staff also who are positive, and we have really dealt a lot about that. There are conflicting papers. There are papers to show that these people eventually land up into some kind of disease later on, but you need to follow up these up.
Yes.
But then it is, some say that is just a normal variation. In fact, there are families with these kind of having these positivities. So they have also been reported. I think the literature is very plus minus, you can't say, but maybe you can follow them.
Super sensitivity of the test is lending us into a problem.
But maybe, sir, it is a time which may be able to, patients may be detected early if we know them. It could also be. We need to have more data on that.
I think we had a case in which the patient was polycythemia vera, and JAK2 was 10%, and then he was started on hydroxyurea. And then he was regularly monitoring with us. We were outsourcing the sample, and it was gradually decreasing. The VAF was decreasing 5% to 1%. I think we just need to monitor such cases, and I think there should be certain guidelines associated. If it is positive, what all we need to monitor the patients clinically for. I think that's what is the answer here. But then again, if it is JAK617F, then it's definitely pathogenic. If it is exon 14, it is definitely pathogenic. But sometimes you do pick up some variants which does not have a clinical significance. They are VUSs, or it is not related directly to any sort of disease. Then you can ignore them. But if it is pathogenic, yes, I think we need to follow certain guidelines for the prevention.
Just one comment again. Hydroxyurea does not decrease JAK2 allele burden.
No, that does not. That does not.
There is a question mark on the lab where you are outsourcing now, where 10% became 1% with hydroxyurea.
No.
Okay. Dr. Radhika has a question. Dr. Nikhil, I'll just come back to you in a minute. Dr. Radhika has a question. She wants to know the difference between clinical exome sequencing and whole exome sequencing. When to ask for which one and what's the difference?
Yeah. So clinical exome is basically a panel which has 8,000 genes, and whole exome is around 20,000 genes. 20,000 genes are the total number of genes which are being clinically routinely expressing the genomic expression involved in transcription and translation. So those are the ones which has been defined, 20,000 genes, whole exome. And when you talk about the clinical exome, that is just a panel of 8,000 genes, which include some of the common, mutations which are related to even neurological conditions and even cardiac conditions, which are most commonly seen in various populations. So the answers to many of these conditions are seen there. So in that also, again, there is a question mark, because even if you go for clinical or whole exome, there are certain neurological conditions like Huntington's disease, which are repeats. Again, for that, you will have to go for fragment sequences. So these panels are basically just to understand the certain diseases which are non-neoplastic and neoplastic conditions. In hematology, we use them because we need to understand if there are any germline part associated with it. And this is how we use it, and sometimes in the pediatric population. Yeah.
So, can I add a comment? So we are using only clinical exome in hematology after the targeted panels are negative. So then most of our clinicians are offering. Otherwise, these are used for neurology and pediatric and other things, clinical and whole exome. But in hemato-oncology, they're mainly, we are using them only for that particular situation.
Germline, yeah.
Dr. Nikhil, yeah, your question.
My question is related to a young MDS patient. So, in these patients, other than getting a myeloid NGS panel, very often we need to work them up for our inherited bone marrow failure. So, sometimes we end up getting a myeloid NGS panel and an NGS for our inherited bone marrow failure syndrome. So, my question is, in this scenario, can we get away with doing a clinical exome sequencing, and would that cover both these panels?
MDS, again, if you're looking at, yes, it will cover, but again, if you're looking at a bigger deletions, which we pick it up on karyotyping and FISH, that might be missed on NGS. And even if it is a targeted panel or it is a clinical exome panel, bigger deletions will be missed. So for that, you need karyotyping or chromosomal microarray or DNA microarrays, yes.
No, my question was regarding whether uh, the genes which are covered in NGS myeloid panel and an NGS panel for inherited bone marrow failure syndrome, are both these covered in the clinical exome sequencing?
Yes.
It's
Mm.
because
Yes.
it would prove to be much more cost effective to get a clinical exome sequencing than to get them both separately.
Yes.
So that was my question.
Actually, just to tell you, Dr. Nikhil, there are many private labs who clinical exome sequencing is cheaper than target exome sequencing for them. So what they are doing is they have filtered the targeted genes and doing the clinical exome sequencing. Even whole exome is cheaper than the targeted sequencing. Only thing is there are some areas which may not be covered that well. So your bioinformatic person has to look into the panel if whatever you are filtering for your hematology panels are covered. So that
Oh,
is one challenge. So that somebody has to look into it. You cannot just blindly go for that. In
so
targeted, you
each
know
panel,
it's
right? So
covered.
each panel is individualized. So we need to go over the genes before sending for the specific test.
Yes, and you at least if somebody you are using, you want a particular gene, and you should talk to the bioinformatic person that just have see if that area is covered and in the clinical, because here there are some 5,000 to maybe 8,000 genes. So they are not bothered for one particular gene.
I think they are covered, ma'am. We have used that panel for many inherited bone marrow
Yeah, most of
samples.
them are covered, but if you're looking for something particular, you
Very
should.
specific.
I'll give you an example of what Dr. Maushmi is talking. We have had some very bad experience, say RBC membrane disorders, then genetic thrombocytopenias. Then recently we burnt our fingers with porphyrias. So, once you get into the rare problems, these labs just give you this clinical exome and they say, oh, it covers everything. And when you're looking at individual genes, they don't exist there. They spend the money and you didn't get the information that you require.
Yeah, because it is not possible to cover everything with same. There you have to compromise somewhere. So where they have compromised, only they can tell you.
Absolutely.
Yes.
Okay. Yeah.
Thank you.
Thank you, Nikhil. Manu, your question.
Thank you, sir. Thank you, Dr. Kela. That is a very nicely organized talk. I'm a hematologist from Sri Lanka, and sometimes due to the availability of resources, we have to send BCR ABL quantifications for different labs. So when we do get reports from different labs, is it possible to compare A and B labs, or if not, is there something we should particularly look for in the reports or communicate with the labs when we are comparing two reports?
I think I always tell anybody, don't compare the lab A with lab B. Always send your sample to one lab and keep on monitoring the same patient from the same lab, because everybody will have its own validation, and it will definitely going to differ. So it is advisable to send this, to monitor the same patient by sending to the same lab, and probably you have checked their validations, you have checked your transportation, that mRNA is not degraded, you have seen that the things are in place, then I think it's best to send it to the one lab. There's no point in comparing the same patient in two different labs.
All right, so probably defer it till it's available from the same lab rather than sending it as well. All right, thank you.
No other raise hand signs. We'll go back to the audience. In APML monitoring, is the quantitative PCR superior to a qualitative PCR?
Qualitative is definitely used as a diagnostic thing, which is used on fish. And quantitative is used on RT-PCR, that is for MRD monitoring. And again, you need to define even initially that what are you dealing with, BCR1, BCR2, BCR3. And once you have defined, you have to monitor the disease when you start the patient on treatment. Yeah.
Thank you. Dr. Kanan Ramamurthy has put a remark on this stroke and JAK2 positivity. Said about there is low-grade positivity of JAK2. In any case, you're going to put all these patients on aspirin. The question is whether to screen for JAK2 in general population, and if positive, to put them on prophylactic aspirin.
Yes.
So, wonderful question. That is what Maushmi was also pointing out too. So that's something probably will happen in 10, 20 years. Dr. Raj Warrior has a comment that panels for the hereditary hemolytic anemia and thrombocytopenia are not expensive and extremely useful. Yeah, probably US, we have a problem here that these labs tell us that the panel covers everything and it does not really cover everything. For the audience, Dr. Raj Warrior is practicing in US. Dr. Mir Hussain has thanked you for a wonderful presentation and clarity of his doubts. Dr. Raj Warrior has another comment. In many non-malignant cases also, NGS and panels have become a better and faster way to diagnose. In NICU and ICU, we are using rapid sequencing to get the result in 24 hours.
Thank you.
Dr. Ambar has a question whether all fusions are covered in NGS for AML.
Again, I think the same question. It depends upon the panel, what you're dealing with.
Dr. Hamza Dalal has asked, can you highlight the principle behind STR analysis for post-transplant chimerism?
Yeah, so what we do is we have initially identified the panels pre-transplant, and then the post-transplant, the patient's blood has donors, stem cells, so which are proliferating. So we measure for the STR fragments for both from the same sample. And if we are able to see the donor STR fragments or the peaks in the patient's blood, and gradually it happens as the complete replacement happens, it goes to 100%. So if you're able to see those peaks, donors peaks in the patient's or the recipient's sample, so this is how we are able to say that the transplantation has happened. But if it starts decreasing, then obviously we are saying that the transplant is failing. If the recipient or the patient, as STR fragments are increasing, so it means the transplant is failing. So this is how I think the peaks I should have shown just to make them understand.
Thank you. Dr. Sutantira Kannadamurthy has another comment. Doing MYD 88 will allow you to use, or you will have a preference to use ibrutinib upfront. He's talking about DLBCL. And then another thing is that doing TP53 will allow you to go for CAR-T instead of a salvage autologous, which is a waste. Of course, you are molecular geneticist, so you may not answer this question, but I do have a remark, Dr. Ramurthy, for that. I hope you're only talking about the relapsed patients, but if you're talking of DLBCL upfront and adding ibrutinib because patient is MYD 88 positive, the outcome has not been great. The other question is from Myanmar. Dr. Senvin wants to know, there's a patient of hepatitis C virus related cirrhosis of liver. Bone marrow shows myelodysplastic syndrome. Do you need molecular studies or NGS panel for such patients?
I think they should first check about the vitamin B12 levels.
Dr. Win, there is always a problem of differentiating dysplasia from myelodysplastic syndrome. So I would say this is a more important indication to go for the molecular studies, because sometimes you have difficulty in differentiating dysplastic changes from an established MDS. So in all such cases, whether drug-induced, virus-induced, as you mentioned about megaloblastic anemia, B12, this, that, when there is a trouble, when you can't make a diagnosis, it helps you. Dr. Raghuvendra has requested you, maybe a subject for the next webinar, give a small talk on history of generation of names of genes. What made them get discovered and how the new genes will be discovered in future.
Oh God.
Yeah, I think that covers most of the questions. So any questions from the people here? If none, then before I hand over to Intas back for the vote of thanks, if they are still around, on behalf of Mumbai Hematology Group, I'm extremely grateful, Dr. Amrit Keller, for preparing this talk, for making it so simplistic, delivering it to us. Exactly, you did what I wanted you to do. And then this long question answer session and interactive session with presence of Dr. Mashmi around, and all the faculty who asked brilliant questions, and the audience who put up so much of comments, and not only from India, but also from Sri Lanka, Myanmar, and US. I think it was worth having these 2 hours. It was great. Thank you, Dr. Amrit. And is Intas there?
Yes, sir, I'm there, sir.
Please go ahead with your official vote of thanks.
Sure, sir. Thank you, sir. We are now towards concluding this wonderful webinar. I must say this was really an exciting program, which has enabled all of us to gain from the wisdoms and expertise of Dr. Amrit Kalar, ma'am. I take this opportunity to thank Professor Dr. M.B. Agarwal, sir, Dr. Amrit, ma'am, Dr. Moshi, ma'am, Dr. Amit Khurana, sir. I also thank to all the participants who have taken time from the busy schedules for the weekend for the celebration part to attend the webinar. And I also thank my interns to providing me the opportunity for the vote of thanks. And due to this color festive season, I wish you everyone happy Holi. Thank you, sir.
Thank you. And really happy Holi to everybody.
Thank you, sir.
Thanks, Dr. Amrit, again.
Thank you, sir.
Happy Holi. Happy Holi.
You.
Bye-bye. Good night.
Bye.